Hi, everyone, and welcome to PRISMS webinar, Understanding the Genetics of Smith-Magenis Syndrome. My name is Allison Stefanok, and I am PRISMS Education Chair, and I'm also the mom to an 8-year-old daughter with Smith-Magenis Syndrome. I'm really excited to see so many of you registered this evening and logged in this evening. Thank you for joining us. This webinar is brought to you by PRISMS. PRISMS is an acronym for Parents and Researchers Interested in Smith-Magenis Syndrome. PRISMS is dedicated to sponsoring research and fostering partnerships with professionals to improve the awareness and understanding of Smith-Magenis Syndrome. PRISMS is also dedicated to providing information and support to families of those with Smith-Magenis Syndrome. So webinars are a new program for PRISMS, and they were developed with the goal of providing information to the SMS community in a dynamic and easily accessible way, directly from the experts working in the field. There is a recording of all of our webinars, future webinars, and past webinars, including this one, and it can be found on the PRISMS website under the Education tab. So at any time during this webinar, if you have a question for our speaker tonight, there is a Q&A box at the bottom of your screen. You can type your question into the Q&A box, and at the end of the presentation, Rachel will take some time to answer as many as she can. She may not get to all of them, but she'll answer what she can. So our topic for this evening is genetics and Smith-Magenis Syndrome. And our speaker is Rachel Franciskovich. She is a board-certified genetic counselor at Baylor College of Medicine in the Department of Molecular and Human Genetics. Rachel works at Texas Children's Hospital in the General Genetics Outpatient Clinic, and she also works in the SMS Specialty Clinic. Thank you so much, Rachel, for being with us this evening. We really appreciate all the work you do with Smith-Magenis Syndrome. We're very grateful to have you around and on our team, and thank you for being with us tonight.
Yes, thank you so much, Allison. I appreciate the opportunity to talk about something that's a passion of mine, and with an organization so wonderful as PRISMS, and something that's very close to my heart. So as Allison was saying, I'm going to go ahead and start by talking about the genetics of Smith-Magenis Syndrome tonight. And thank you everyone for joining me. So give me one second. Okay, so just to give everybody a little bit of background about who I am, so that you know that I'm not just somebody talking to you about genetics, because I like it. So I got my bachelor's degree from the University of Houston here in Texas, and then I got my master's degree in genetic counseling at Ohio State University, so I've done a little bit of traveling here and there. But my first actual real experience with Smith-Magenis Syndrome was because I was a student intern in Dr. Sarah Elsey's lab, and I'm sure many of you all are familiar with Sarah Elsey, as she's one of the Professional Advisory Board scientific advisors, and she does a lot of work with PRISMS itself and doing the research. And so my internship in her lab sparked an interest with the families in the community surrounding SMS, and gave me that launch pad and diving board basically to get into this world and provide the families with even more information than I even knew was available to give from myself. All right, so let's go back to school. It's that time of year again, and all of us at some point in time have had some kind of genetics, whether we remember it or not. But I wanted to start with just a basic genetics lesson, so that when I start talking about the nitty-gritty of Smith-Magenis Syndrome and what the genetics mean, everybody has a kind of context for what that looks like. So to start off with, this is just basically a graphic showing the very broad basics of what we're going to be discussing today. And so as you can see, there are we start off with one cell in our body, and we are all trillions and trillions of cells. So we make up each cell makes up our genetic instruction manual, and inside that instruction manual, you should have your chromosomes. And then the chromosomes are basically the DNA and the genes that are all one huge instruction book that we use to understand our bodies. So on your screen is what's called a karyotype or a chromosome analysis. And what this is basically the genetic snapshot of our chromosomes. Somebody much smarter than myself, I say, in a lab, takes a look at one cell of our body and opens it up and organizes these chromosomes 1 through 22 and X's and Y's. So you can see here we have one copy from our mom, one copy from our dad of each chromosome. So we have 46 total, 23 from mom, 23 from dad. There's males, who down here in the bottom would be X and Y. That's the only difference between men and women have 2 X chromosomes. Something that's important to note in particular when we start talking about specific locations and coordinates for Smith-Magenis syndrome and many other genetic conditions for that matter, you have what's called a P arm, which is the short arm of a chromosome, the top part usually. And you have the Q arm, which is the long arm or the bottom part of the chromosome. And like I said, that's more important when we start talking about where the chromosomes are and what their importance is. So back to this picture, we've taken a look at the cell, which is the biggest piece, zoomed in and taken a look at the chromosomes. And when I refer to the centromere up here at the top, that's where the split happens between the P arm and the Q arm. And then if we zoom in even further, we can see the genes or DNA. So the biggest question that many of us have asked ourselves before or may have a vague knowledge of is what is DNA and what does that mean? So DNA stands for deoxyribonucleic acid, which is just a very fancy word for saying it's the most basic building block of who we are. We all have our DNA that makes each one of us different and special, and it provides instructions for things like our hair color, our eye color, what color our skin is going to be, what our instructions are for chances to have certain diseases, anything that you can think of, your DNA in terms of your personality and things like that is the instructions for that. Then we get even further into the nitty-gritty, and you think of the building blocks of DNA as A, C, T, and G. And what I say these are basically our genetic alphabet. So if we're trying to spell words, the only letters that our genetic alphabet is going to use are A's, C's, T's, and G's. A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. And as you can see here in this picture, A always pairs with T, and C always pairs with G. These are just the way that our DNA is instructed to match itself, and it's based on 2 strands. So the top strand as A gives us the bottom strand matches that with a T. Here we have the extended DNA. So this is basically us looking at our alphabet, and we start seeing that we have a bunch of letters that are matched up, but what do they even mean? So each 3 pairs of our DNA or our alphabet pair up together and are read, basically like a sentence, to be a certain amino acid. That amino acid is dependent upon what 3-letter code that the DNA has given it. And here I have a variation of what some of you may have seen before in some of your children's or individuals' clinical genetics report, depending upon what they have. And it's basically just saying that the instruction starts with what amino acid is there and how it builds it up. So at the very top here, the reason that the previous one and the top one here in the MET or MET, which also stands for methionine, is pink, is because our DNA starts with methionine, and that's where it starts reading our DNA and our instruction manual which are the sentences or the genes. So when our body's DNA is read through the instructions, it hits that 3-letter code. It says, start reading here. Basically, if you or I were to read the start of a sentence that said start. As you go, these amino acids make up a chain that then creates a protein or genes that we can see that are the instructions to tell our bodies what processes are about to happen or how we are supposed to understand the instruction manual that we've been given. This can be things like your instructions for how your body is supposed to process fats. It can be your instructions about how your body is supposed to process the colors that you see through your eyes. So if we put this all together, we can think of this as a very large instruction manual where we start with the genome or the karyotype that I showed you originally, and that's the cover-to-cover instruction manual. Then you take it further and you think, okay, to read an instruction manual, I have to read chapters or sections. If we think about all of us opening an instruction manual to build something like a desk, they break it into sections to build pieces. So our chromosomes are those chapters. Then you move forward and you can't just read a chapter to build the desk. You have to know which section or which sentence to read first and how to read that. And you can think of those as your genes. So this is something where if we're thinking about genes related to Smith-Magenis syndrome, the most important one is called RAI1, and that sentence is important for understanding what Smith-Magenis syndrome can do. You go even further down and even closer on your microscope when you're reading, those sentences are made up of words. And those words are similar to what I had said with the amino acids. They start the building block of those sentences to give us our instructions. And finally, if we go back to the very beginning, the DNA is the alphabet that makes up everything in that instruction manual, and the alphabet is A, C, T, and G. Okay. I know that that was a very quick and dirty run through of high school genetics and biology, but hopefully we all have a little bit of an understanding of what I even am talking about when I start talking about the genetics of the specifics of Smith-Magenis syndrome. So as you're aware, or maybe not aware, Smith-Magenis syndrome is caused by typically a chromosome 17 deletion. So think of it here as typically you would see 2 chromosome 17s that look the exact same in every single cell of a person's body. And again, we have trillions of them. But from the moment that a cell is fertilized and made into an embryo, this chromosome, for some reason, just by chance usually, got a piece that got cut out of it. And so as you can see here on the right of your screen, at least I hope it's the right for you guys, is what the chromosomes would look like if the individual is missing the piece that is related to Smith-Magenis syndrome. When we in genetics read coordinates for genetic conditions, in particular chromosome conditions, I like to think of them as their GPS coordinates, but we just read them really funny. So for you guys at home, you usually would read where Smith-Magenis syndrome is as 17p 11.2. And for us in genetics, we try to be fancy and confuse you even further. And so we call it 17p 11.2. So if you hear somebody saying that, they're still referring to Smith-Magenis syndrome. Smith-Magenis syndrome is considered something called a contiguous gene deletion syndrome, or in easier terms for me to understand even myself is to say continual gene deletion. As you can see here, we have a chromosome 17, and we've taken and zoomed into the part of 11.2, and this is a very broken down picture, easy picture to look at. we can think of the genes as A, B, C, D, E, F through J here. And that's the normal, typical pattern for what chromosome 17 should look like. However, on the other copy of chromosome 17, which would be the one that we're considering for Smith-Magenis syndrome, you can see here that C through F are no longer there. So we think of this as the deletion of multiple functionally unrelated genes, which one copy is deleted from. Functionally unrelated, meaning that the instructions or the sentences that are contained within this section that's deleted don't perform the same task and need to do multiple different jobs for our body to understand its instructions. Another term that you may have heard of before is something called haploinsufficiency. That means that we only have one amount of the C, D, E, F instruction in all of the cells of the body. And because of that, our instruction manual no longer makes sense. And for individuals with Smith-Magenis syndrome, that instruction not making sense to them is what causes many of the common features that we can see for these individuals and the differences that we can see. So you do have a copy on the other chromosome or the other instruction that is doing the job that it's been told to do by its instruction manual, but because it doesn't have its partner, it doesn't quite make sense. Something that I've mentioned before is that the typical deletion that causes Smith-Magenis syndrome is that on P11.2, and it contains about 80 genes, which our body has 20 to 25,000 different genes in our body. And we only know about probably 7,000 to 8,000 of them do at the current time. So that you can, as you can imagine, there are a lot of things that we in genetics don't understand. And this deletion is usually about 3.5 megabases, which is basically 3.5 million base pairs, which is those DNA letters. So there's a lot of instructions that are missing out of the instruction manual when we're thinking about Smith-Magenis syndrome. Other causes of SMS is not just this deletion. There can be something called a pathogenic variant in RAI1 or some kind of different alternative deletion size. So in talking about RAI1, this has been identified as one of the causative genes, or basically the gene that we know causes many of the symptoms that we can see related to SMS. And it's in the example that you see on the screen is of a pathogenic variant or a base deletion in the gene that can cause SMS. In the genetics world and in the way that our body reads the sentences for genetic information, that DNA is supposed to be written in a very specific pattern with only a few minor spelling changes here and there that make each one of us individuals, why we have different colored hair or different colored eyes. We're prone to different kinds of genetic conditions in our families. But sometimes when our body makes those spelling changes as they normally would, it can cause the instruction not to be read correctly, and it's an important instruction in our body. In the case of Smith-Magenis syndrome, that important instruction is RAI1. And if it's got a spelling change, that usually occurs about 10% of the time to cause the symptoms that we can see with SMS. Okay, so these are the very basics of SMS as a whole and what are the different causes that we can see related to SMS genetics. So that's the deletion of 17p11.2, or some children have been diagnosed or adults have been diagnosed by a pathogenic variant in RAI1. Many families this diagnosis has been known for a number of years, and in genetics, our technology constantly improves and constantly changes. So there are many, 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 many different ways now that an individual with Smith-Magenis may have been diagnosed. And I think it's important to go through and understand what each of those could have been and why each of them is just, it doesn't matter which one has been diagnosed, it's just helpful to understand the techniques that were used. So early in the genetics world, the way that we were looking for genetic conditions and being able to understand the instruction manual or the karyotype or the chromosomes was by something called FISH analysis. FISH stands for fluorescence in situ hybridization, which are just big words to basically say that we stain or paint parts of the chromosomes that we know about to be able to look at them under a microscope and understand what we're looking at. In this first image, you can see the not deleted FISH. They had painted the chromosome 17 with the centromere and point, and they've painted the end of chromosome 17 to be able to know what we're looking at. So in the first image with the not deleted, you can see we have two green points where there are dots lit up and two orange points where there are dots lit up. And that's telling us and telling the person looking at the chromosomes that we see that the chromosome 17 is as it should be in a typical cell line. Then on the other side, you have the deleted FISH result, which basically means that, as you can see, we see one section that has the green lit up for chromosome 17, but on the other one where the orange should have green as well, there is no 17 green lights or green painted spots, basically telling the person that individual in that cell line is missing the instructions for that piece of chromosome 17. This analysis is not very accurate in telling us what is involved in that deletion. And so many times, if we have an older adult that has come in with a diagnosis of Smith-Magenis based on FISH results, we typically suggest performing additional testing to be able to understand just how large or small that deletion is to know what to expect. But this is something that was done very early on for many of the individuals that were diagnosed. Next, you have something called comparative genomic hybridization, or what many of us call a chromosome microarray, or CMA. This technique measures the DNA copy number, so we should have 2 copies of each amount of DNA in our body by comparing it to a control sample to the individual that the test is being run on. So every test that is run through for a CMA has a healthy control or a healthy individual running next to the individual that is in question. In particular, for those with Smith-Magenis, many times this test is run under the question that this child or individual has some type of developmental delays or different facial features than we would expect to see. So there are different kinds of arrays that can be used. Some are meant to target specific syndromes. However, most of what we do in today's genetics clinics are called a whole genome array, which basically covers all of the genome that we can get, and there's millions of these probes on each basically chip for a CMA, and millions of different places that we can look at and tell us where a deletion starts and where it ends. The resolution is much, much better. Basically, the way that we can look at it at the lab is much better than we can do just by looking at either a FISH or that karyotype, which that karyotype was the very beginning picture that I had showed of our chromosomes. And it's very difficult many times to look at that snapshot of our chromosomes to be able to find deletions such as Smith-Magenis syndrome or other genetic conditions because they're actually too small to see on that snapshot. And so, as I said before, this is the most common method that we see in the genetics clinic for a diagnosis of Smith-Magenis syndrome, because it is recommended as one of the first tier of testing for any individual that has any kind of developmental delay or multiple birth defects. And so this is typically something that especially newer diagnoses in the SMS world would have received. So this is something that I thought was going to be of interest to some people because it never actually gets to you guys on the family side. We usually just hand over a report that reads off that the individual that was tested has a 17p11.2 deletion. However, when this is actually being done in our labs, they are looking at the probes that I was talking about, and they're looking to see they all should be at that 2-copy number, and that's considered the 0 for many of these individuals, so that's the middle line. And we're looking to see, are there any extra pieces, which would be the top, it would move towards the top of your screen, or are there any missing pieces, which would move down towards the bottom of the screen? And as you can see, the numbers on the far left there go from 0 to 3 up top and 0 to -3 at the bottom. And the individual that was tested for Smith-Magenis syndrome on this CMA in particular shows that at that point of P11.2, they had 1 copy less than we would expect to see for chromosome 17, making that diagnosis. You zoom in even further, and you can see at the bottom here that there is a large chunk that was moved down by 1 piece of the chromosome itself. And when we evaluate these in the lab in particular, we look to see what instructions can be contained in this amount of genetic information. And if you remember, I said that there are about 80 different genes in the common deletion, but that we have 20 to 25,000 different genes in our body. So if you imagine missing a very large part of a chapter book and you're trying to put the top of a desk on and don't know how to do it, then you never get the top to your desk. So as you can see here at the bottom of the screen, there's RAI1 that is contained in this region. And as well, something that you may have heard of or heard about and would not be addressed in this webinar, but a mirror condition is Potocki-Lupski syndrome, because that's the extra piece of the same genetic information that individuals with Smith-Magenis syndrome have. They just have an extra copy instead of a missing copy. And then you can see down here at the bottom, as we progress through this, that there are a large amount of genes, or basically a large number of sentences in that instruction manual that are contained inside this 17p 11.2 GPS coordinate. However, RAI1 had been found out to be the most important gene related to what we can see with individuals with Smith-Magenis syndrome. This is just an easier representation to understand why and how we understood this information to be. And so if you zoom in the labs, they were looking at, we had many, many individuals that had Smith-Magenis syndrome. We know that they all had the similar characteristic features that we are aware of, like the circadian rhythm, flip. They have some similar facial features to each other. And they started to wonder what was causing these individuals to have some of the same presentations in clinic or same symptoms. And so many different deletions were looked at and were worked with and targeted down to where the smallest region that overlapped must have been the most important piece to cause Smith-Magenis syndrome for our individuals, and that was considered to be the critical region. Within that critical region was the RAI1 gene. And as I've talked about before, you can see individuals with a pathogenic variant or a spelling change in that instruction. So many times I'm asked, or we are asked as a team in genetics, so was it due to just one gene to cause Smith-Magenis syndrome, or are there many genes that cause Smith-Magenis syndrome? And as I've talked about before, many of the features that we can see are related to RAI1. However, it still is important to know that there are other instructions or other sentences that are missing in SMS, and that each and every one, even if we don't know their exact importance, must have some kind of information that is important to that individual's instruction manual. So if you have a child that only is missing genes C and D plus RAI1 versus a child that's missing A through F in RAI1, they may very well have the common things that we see for SMS, but the one that has a smaller deletion versus the one that has a larger deletion may not present the exact same way because their instructions are slightly different, just like every single one of us have slightly different instructions. As I've talked about a little bit before and wanted to touch on a little bit further, we all have our genetic instructions with our DNA. And when we are looking at RAI1 in particular and that pathogenic variant, because I've talked about how an individual would be diagnosed with a CMA, in individuals where we are trying to diagnose and we think that they have Smith-Magenis syndrome, but their chromosome microarray doesn't tell us that they have a deletion, our next question is to say, okay, is their RAI1 gene spelled the way that we would expect it to be spelled? Because we know that RAI1 causes many of the symptoms that we see related to SMS. And so here you can see at the top that as we read through, basically, again, somebody I say that is much smarter than I am, goes through, and the middle picture is what a readout of our genetic sequencing would look like in software that is used. And at the top of that, you see that it's actually spelling out the letters that you would use in your DNA. And for this individual, there is a missing C on the deleted part or the second copy of their RAI1 instruction. If you remember from the beginning of the talk, when I was talking about the amino acids, every instruction in our body is read as 3 letters of our DNA grouped together. And as we're reading through that, you can imagine if you delete entirely one of those letters, then your new instruction is going to read with that next letter and the 2 after that, which typically causes that instruction in our body to not process it correctly and make either a different protein, a different amino acid that causes something not to work entirely, or it can cause less of a certain protein to be made in our body, which can cause clinical symptoms. And so this is a common second tier or second step in testing for individuals to be diagnosed with SMS if the CMA is not diagnostic originally. And so this is something that can be difficult for families in particular because each test obviously requires blood draws and turnaround times, and can be difficult for families to wait for the diagnostic odyssey when we know what's going on, because we always start with that first tier in testing, and that takes time to come back with the CMA. And if we think, okay, this individual we feel like they have Smith-Magenis syndrome, as we commonly do in genetics, then we have to wonder and ask to sequence the gene, or basically read through that gene to be able to understand if that individual truly does in fact have SMS. So many people ask that if we're so commonly diagnosing SMS based on the deletion, how do we know what percentage of individuals have a pathogenic variant in RAI1? And we estimate that that is about 10% of the SMS population, or the group that we do know have a clinical diagnosis of SMS. But I would say that it is likely underdiagnosed. Until recently, RAI1 was not included on many broader panels for genetic testing panels for individuals with developmental delays, or what's considered syndromic intellectual disability, where we look at an individual and we say, while they have intellectual disability, we also think that because they have other symptoms, they seem to have a genetic syndrome. And it is also not until fairly recently that RAI1 has been included on genetic testing panels for things like epilepsy, and for things like obesity panels, because this is something that we know for individuals with RAI1 variants, they can have epilepsy or seizures that are more common, and for obesity, that there's many individuals with SMS that can have rapid weight gain and obesity that is uncontrolled without intervention. So the question remains, and I've kind of touched on it here and there, is RAI1 the only gene, or are there other genes in that 17p11.2 region that are modifying this disorder? So what we know and what we've looked at is that a mutation or a pathogenic variant is another, mutation is another word that you guys may have heard, or even a deletion of RAI1 results in many of the features that we see in SMS. However, as I've touched on before, and I think is important to know, is that some genes contained within the deletion are important to have for that individual with the 17p11.2 deletion, because our genetic background is what makes each of us who we are. And so it's something that we consider, or we call in genetics, variable expression. And every individual with SMS has very common things that go along with it. Many families tell us about the sleep struggles, and many families tell us about the behavioral difficulties, especially as children get older. But not every individual with Smith-Magenis syndrome has the same clinical things that they're going through, and that's because of potentially what the genetic background is and what's made up of the instructions that are contained within their missing piece. So many families come to us when we're talking about the diagnosis of Smith-Magenis syndrome, and we say, okay, we know how it happened, we understand how our child was diagnosed, but will it happen again? And I think that this is such a normal question to ask, and such a normal question that we ask ourselves, especially for many families. This diagnosis is made sometimes as very early on, as early as like months old, but sometimes in the 3 or 4 year old range. And a lot of families, if this is their first child, want to know what the chances are if they're going to have another child with SMS. And so that's many times where you will find yourself in a genetics office with someone similar to myself, a genetic counselor, and we go through the same information, a lot of what I've talked about. And so thinking about genetic counseling from top to bottom, kind of going who, what, and how. As we said in this, we're talking about Smith-Magenis syndrome, and we've talked about throughout this webinar what caused it. And so that's commonly, roughly 90% of the time, it's caused by that deletion in chromosome 17p. But there's also that roughly 10% or potentially greater that have pathogenic variants or mutations in RAI1. And the next question that we always get asked is it inherited? And I think that it's important at this point to distinguish that inherited and hereditary are commonly used interchangeably, but they do not mean the same thing. And so inherited is something that you can see running through multiple generations of a family, and you can see mom has one thing, she inherited it from her father with the same exact genetic diagnosis. But this is still considered hereditary because it is included in their instructions. However, this is not commonly something that we would consider inherited. Most all of the cases that we see are considered de novo events, or basically what means sporadic or by chance when that fetus, when that embryo is being formed, that missing piece did not get packaged in properly. So it means that the genetic cause for SMS was a random event, and most parents are not considered to be carriers or are not considered to harbor the deletion themselves. However, that being said, it's not always that easy in genetics, and we're never going to tell you never, because the minute that we tell you never is the minute that you show up in our clinic saying it happened again and how. So in any child that is diagnosed, or any newly diagnosed case of Smith-Magenis syndrome, we will always, 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 always recommend for the parents to get tested as well. As much as I can tell you that the chances are very low that a parent will not be a carrier for this, there are certain types of chromosome rearrangements, which basically means that pieces of chromosomes in your instructions are in the correct number, but that when you go to have kids, when the chromosomes rearrange themselves and a missing piece is packaged in because yours are in the correct number, but you have a child with a missing piece. These are extremely rare, and frequently we can pick them up, but after some very good detective work on a few genetic counselors and geneticists' part, because it's not very clear cut and straightforward on how to do so. So this would be something that each individual case would need to be managed separately. Something that I feel like I get asked more frequently, especially more recently, is what about mosaicism? And so it's hard to even explain mosaicism when you're not in front of anybody, but it's something that I would say is considered that you have two instruction manuals contained inside one body. So I like to think of this as tiles on a wall, and if we all were to put tile backsplashes up, it'd be something that commonly, if you're trying to create a pattern, you're going to want some white tiles and you're going to want a black tile in the middle. Say we're making a square around a black tile. And however, there are all of those of us that would just prefer a plain white backsplash in our wall, tile backsplash. If you have a plain white backsplash, you shouldn't have any mosaicism or any different cells or instructions in that backsplash. But if you think about the black backsplash, there are random places on that backsplash that have a different instruction manual. There are two types of mosaicism. The first one being something called germline mosaicism, which means that there is a normal cell line running in that individual's sperm or egg, depending on the parent. And there is also a cell line in the sperm or egg that could contain the deletion of 17p or even just an RAI1 pathogenic variant. These are the kinds of mosaicism that we cannot diagnose because we are not going to be going invasively taking sperm samples or doing an egg biopsy to determine this. And so this is why we never say never, because we can never be 100% sure that there's not germline mosaicism occurring for that individual. The other kind is when some individuals are diagnosed with Smith-Magenis syndrome, either on the CMA or when we're reading through their genetic instructions for RAI1, that the report tells us that they have mosaic SMS, or basically what means that when they looked at some of the cells, they didn't contain the deletion or the pathogenic variant, and when we looked at others, they did. This occurs in a low number, about 3 to 5% of SMS cases, and it can change the recurrence risk for the family. So this is a visual representation of what mosaicism looks like and could potentially help explain visually what I was trying to say. And so as you can see here, for what's called somatic mosaicism, which is different than germline mosaicism. Somatic mosaicism is any cell in our body that is not our egg or sperm cells. And so you can see that every cell has to split and copy itself over and over and over and over again to make up the trillions of cells in our bodies. And as it's splitting and copying itself, by chance, one copy of the cell gets a deletion or gets a pathogenic variant in RAI1, and then that continues to copy itself, but separately from the copy that didn't have any problems to begin with. And so as you can see here in the visual representation, it basically means that some instructions in our body have one set of instruction manual, and the other part of our other set of instruction manual in our body has the instructions related to SMS. Germline mosaicism, the visual remains the same. However, this starts with egg or sperm because the instructions are different there, different cell type there. And this is something that we cannot test for. And this is what is altering that recurrence risk for many families. So to give you an idea of the recurrence risks, for many parents of a child with SMS, and we go through the parental study testing by either looking at a parental karyotype, parental FISH studies, parental chromosome microarray, or in the event that the child is diagnosed with an RAI1 variant, we do very site-specific testing and find out that neither parent carries that. Then the recurrence risk is low. We usually tell you it's less than 1%, because like I said, genetics, we can never say never because we've all been surprised. In that case, for future pregnancies, prenatal testing is available for what we would consider quote unquote at-risk pregnancies, but basically to say for a family history of SMS, there is prenatal testing available. And many times on the prenatal side of things, they would use an SMS-specific FISH probe. So basically, like I said at the very beginning, they would paint the chromosomes that they got from the fetus that is growing, but this would have to be an invasive test, so something called chorionic villus sampling, CVS, or an amniocentesis, that is the needle test for many people are aware of. They can also do the chromosomal microarray on the cells from the fetus, or if needed, site-specific RAI1 testing. Frequently, we have families that have their parents, but then they have other children and want to know what are the chances that my other child is going to have a child of their own with SMS. And again, if the parents have normal chromosomes, then the risk is, again, very, very low for their siblings' children to have SMS. And then the question remains, what is the chance that my child with SMS will have a child of their own with SMS? And there is, because this is considered a dominant disorder where it only takes one copy of chromosome 17 to not have that information to present symptoms of SMS, it's a 50-50 chance, similar to like flipping a coin, as in each pregnancy, whether that individual would have a child with SMS or not. And here we say it's a theoretical risk because it depends upon the level of intellectual ability for the individual with SMS as to whether or not they would be able to have children of their own. And that's an ongoing conversation that we would be continuing to have in the genetics clinic. So I hope that you have had a blast back to high school biology with me and have a little bit of a better understanding of the genetics of SMS. And I am happy to answer questions as we go through them.
Thank you so much, Rachel, for taking us all back to school and teaching us about genetics and the genetics of Smith-Magenis Syndrome. We really appreciate it. We have some time for questions. We have some really great questions for you, as I'm sure you could expect with your awesome presentation there. Okay, so let me pull up questions and we will go through as many as we can tonight. We might not get to all of them. Let's take a look. So the first question for you is, are there studies for older SMSers? Is the syndrome most prevalent in the UK and European countries?
Yeah, so that's a good question. There are, if you're referring to studies like research studies for older SMSers, I know that we have the PRISMS registry that is ongoing that is going to allow us to open up some of the older diagnosed SMSers into the research world to be able to understand further. But there is no difference in the prevalence of SMS in other countries like the UK or European countries. We, based on the genetics, there shouldn't be any difference in those individuals being diagnosed here in the United States versus those in the UK versus those in Europe versus those in Africa. It should be the same everywhere. And that's just based on it being a deletion of a chromosome.
Okay, thank you for that. Next question is there any difference in symptoms for a deletion of RAI1 versus a mutation?
So this is a little bit tricky to answer. I would say that the deletion of RAI1, if it's contained within the large group of 17p11.2 versus a mutation, it should not change many of the symptoms. They are going to vary slightly, and that more so has to do with the fact that an individual with a deletion containing RAI1 has more genetic information that is missing versus somebody that only has a mutation in RAI1.
Okay, thank you. Our next question is, we originally had a microarray in 2016 that did not catch Smith-Magenis. It did, however, show a chromosome 5 deletion. Is this related to SMS?
So that's a really great question, because I think that frequently if the chromosome microarray has been done and did not identify SMS but did identify something else, it does not typically relate to a diagnosis of SMS. So a chromosome 5 deletion still has important potential impacts for that child or individual's care if there's important instructions there, but it would not be related to SMS.
Okay, thank you.
Mm-hmm.
Our next question for you, Rachel, is in your discussion of Mosaicism, you mentioned sperm and egg. Mm-hmm. Is this an SMS individual, and the de novo occurrence is one parent more prevalent as the cause for the de novo activity causing SMS?
Not 100% sure that I understand how the question is worded or being asked. But in mosaicism, typically there's not one or the other that would be more likely, sperm or egg, to have a change. However, that being said, sperm is more common to have de novo, basically pathogenic variants or mutations in it, just because of the vast number of sperm that are produced for males versus the female's eggs. So hopefully that kind of answers that question.
Okay, thank you so much, Rachel. Okay, I think we're coming to the end of the hour. I think that's all the time that we have for questions this evening. I hope that everyone enjoyed this evening's presentation on the genetics of Smith-Magenis Syndrome. Remember that a recording of this webinar, as well as all future webinars, and a list of our upcoming webinars and recording of our past webinars can be found on PRISM's website, prisms.org. And also, please be on the lookout for an email from me with a link to this recording as well as a survey for this evening. Please take some time to fill out that survey if you get a chance. We'd love to know how we did this evening. I would also love to see all of you on future PRISMS webinars, and please be sure to follow us on Facebook and join PRISMS if you haven't already done so. Membership is free, and you can sign up for that on the PRISMS website as well. PRISMS is a nonprofit organization. All of our programs are solely funded by our generous donors. So if you would like to make a contribution to support the programs that PRISMS puts out, then please visit our website again, and you can make a contribution there. And this concludes our webinar for this evening. Thank you so much, Rachel, and I hope that everyone has a great evening. Bye-bye.
Thank you.